Welcome students. To today's session, we are doing the topic separation of substances, and we have been studying various methods of separation. Today we will be doing the method known as chromatography. This method is used for separating colored components from a liquid by using a filter paper or a blotting paper. This method can be used to separate colors in a dye, pigments from natural colors, and drugs from blood. That means chromatography is used for analysis of blood and what sort of drugs are present in the blood. Pigment chlorophyll contains four different colored pigments which can be separated using this method. You can separate what and all colors are present in your blue and red ink. Now, while performing this experiment, we have to put a drop of ink near the end of a strip of filter paper. You can try this at home. Put a drop of ink at the end of a strip of filter paper and dip the end of the paper in a test tube containing water. Ink is actually a mixture of two or more colored components. These components, which are more soluble in water, rises faster and get separated. So the strip is put in this manner, and you can see that the black ink is made up of so many different colored components. You can see there is yellow, there is purple, there is blue. So this can be separated using this technique of running the dye in water. So the different colors are separated very beautifully. You can see the insect picture, how the colors get separated. Let us move on to the next topic. The next method is distillation. This method is used to separate a mixture of miscible liquids by boiling the mixture and cooling and condensing the vapors. There are two different types of distillation, simple distillation and fractional distillation. Simple distillation is used for separating a mixture of two miscible liquids, having sufficient difference in their boiling points. Example, if a mixture of acetone and water is taken, how we will separate it? It is using a simple distillating column. The mixture of acetone and water is heated in a distillation apparatus, which is seen in the picture. The acetone, which have lower boiling point, evaporates first and comes into the condenser. The condenser is run with water, which cools the liquid, and you can collect the acetone. What will remain back is water, which has got a boiling temperature of 100 degrees Celsius. So acetone comes out earlier and it condenses and it can be collected out. So this is a method which is called as simple distillation. Now let us look into what is fractional distillation. Fractional distillation is a method used for separating a mixture of two or more miscible liquids whose difference in boiling point is less than 25 Kelvin. Now, in this, the apparatus contains a fractional distillating column, and a fractional distillating column is designed in such a way that fractions of the material get separated out depending on their boiling points. The apparatus the apparatus used for fractional distillation is similar to that used for simple distillation, except that a fractionating column is fit between the distillation flask and the condenser. The fractionating column has glass beads which increase the surface area for the vapors to cool and condense. Fractional distillation is industrially used to separate components of petroleum and the different gases from air can also be separated using this fractionating column. So the apparatus is over here. Different fractions separate out and they can be collected at different temperatures. So this is called fractional distillation.
Let us see how we can separate the components of air. Air is a mixture of gases. The components of air can be separated by fractional distillation. Air is compressed by increasing the pressure and cooling by decreasing the temperature of air to get it into a liquid form. The liquid air is then allowed to warm up slowly in a fractional distillation column. Then the different components are separated at different heights by increasing the temperature depending on their difference in boiling point. This is a flow chart. You can just go through it. Air is compressed and cooled by increasing pressure and decreasing temperature, and air is converted to liquid air. This is allowed to warm up slowly in a fractional distillation column, and the gases get separated at different heights depending on their difference in boiling point. The boiling points of the following gases are: oxygen is minus 183 degrees Celsius, argon is minus 186 degrees Celsius, and nitrogen is minus 196 degrees Celsius. The next method of separation we are going to study is crystallization. Crystallization is the process of obtaining pure solid crystals from its solution. This is the method which is commonly used in getting our common salt from seawater, crystals of sodium chloride. By crystallization, we can obtain pure copper sulfate from its solution. We have to dissolve around 5 grams of copper sulfate in minimum amount of water. We have to filter the solution to remove the suspended impurities. Evaporate the solution in a china dish to get a saturated solution. Cover the solution with filter paper and allow it to crystallize. Pure copper sulfate crystals are formed. You can have a look at the pictures given below. The difference in color in impure copper sulfate and pure copper sulfate. So with that we end today's session. Thank you.